Welcome, everybody. Today we're going to be talking about motility testing, specifically the STAB method. Let's start off by what is motility. Motility is the ability of organisms, cells, or microorganisms to move independently using metabolic energy. The ability of bacteria to be mobile relies on whether it has flagella or not. Flagella are microscopic hair-like appendages that extend from the cell membrane of prokaryotic and eukaryotic cells, primarily serving as specialized organelles for motility. Bacteria that possess flagella are motile, while those that do not are called non-motile. What do we need to perform a motility test, and what are the steps? For a motility test, we need a bacterial culture, a motility media, and an inoculating needle. For motility testing, we use a specific type of media called semi-solid media. This media is made of about 0.5% agar compared to the standard 1.5 to 2% in solid media. This reduction in agar concentration allows motile bacteria to move away from the stab line. After an incubation, the tube used will show either a clear growth of bacteria along the stab line only, which hints to non-motile bacteria, or it will demonstrate a spreading growth away from the stab line, indicating motile bacteria. Now let's get into the steps of our test. First, we prepare the motility medium. We use a semi-solid agar deep tube for this test. First comes the sterilizing of the inoculating needle. We use a Bunsen burner to flame the needle and then we let it cool down. Then we pick a bacterial colony. Take a small amount of the test organism using the sterile needle. Usually, a small touch is all it takes. Stab the medium. Insert the needle straight down through the center of the medium and avoid touching the bottom. Touching the bottom could result in a false positive result. Pull the needle out in the same straight line. Then comes incubation. We observe the result after the incubation. Motile bacteria will show growth that spreads away from the stab line, as shown in the left tube, while non-motile bacteria will show growth that remains only along the stab line, as shown in the right tube. Now let's get into our demonstration of this test.